Thank you for watching our Teaching the Genome Generation webinar about our new content focused on the power of data analysis for understanding human health. During the webinar, I will start with a brief introduction to our Teaching the Genome Generation program, provide an overview of the new curricula, and then discuss the two new modules, one on health disparities in cancer and a case study titled Precision Over Presumption. For anyone who is not familiar with our program, I will first share a bit about who we are. The Jackson Laboratory is a nonprofit research institution focused on discovering precise genomic solutions for disease and improving health. Our genomic education department delivers modern learning experiences focused on genetics and genomics to both internal and external audiences. Our Teaching the Genome Generation project, TtGG for short, is a training program for high school educators, allowing teachers to bring curricular resources and scientific experiences into their classrooms for their students, all for free. We believe today's youth are the genome generation. We are focused on serving today's youth via the educators that support their learning. Our program aims to make genetics and genomics accessible to students and teachers. We provide a modern approach that links classroom content to personalized medicine, ethical decision-making, and everyday life. We see genetic variation as a cause for celebration, the differences that make us each unique. With our curriculum, we hope to illuminate the relevance of genetics to everyday lives and celebrate human genetic diversity. Our program has been supported by Science Education Partnership Awards from the NIH. We are first and foremost a high school teacher professional development program. We train educators in our human genetics laboratory curriculum, focusing on high-needs schools and districts in New England, local to us, such as urban New Haven and Hartford in Connecticut, and rural areas of Maine. We loan lab equipment and give reagents to trained teachers at no cost to them. Our second SEPA opened training to pre-service teachers and allowed us to broaden our curriculum by adding data science-focused lessons into the genetics curriculum and biology classroom. A supplement award to our NIH grant in 2023 supported the project we're debuting here, developing new curriculum focused on health disparities, social determinants of health, and precision medicine. Our goal for this supplement award was to develop curriculum at the intersection of genetics, health disparities, and social determinants of health. This process had two steps. First, conducting a needs analysis by doing focus groups with a variety of teachers, and then developing curriculum based on the outcomes of the focus groups. We met with 3 different focus groups representing the range of teachers and schools that we work with, a group from rural Maine, a group from suburban Connecticut, and a group from Memphis, Tennessee. During our conversations, we solicited feedback about the content topics, skills, and lesson formats that would work best for the teachers and their students. Incorporating the feedback, we developed the 2 modules we are debuting today. In the Health Disparities in Cancer module, students explore cancer data from the National Cancer Institute SEER Explorer and survey data from the National Institutes of Health All of Us research program to construct explanations about the association between social determinants of health and cancer outcomes across populations, with a focus on the distinction between correlation and causation. The Precision Over Presumption case study introduces concepts within pharmacogenomics and population genetics using allele frequency data from the All of Us research program. The case is framed with the story of a neurologist and their patient who suffers from migraines. First, I will walk through the Health Disparities in Cancer module in more detail. This module aims to engage students in thinking about 2 essential questions. How do social determinants of health impact disease risk, prevalence, and outcomes? And how can we use data to answer questions about human health, health disparities, and social determinants of health? We hope they will come away from the module with the enduring understanding that health disparities are driven by social determinants of health, not by biological differences. Through the activities, they will utilize their skills in data analysis and interpretation, asking questions and formulating hypotheses, pattern recognition, and constructing explanations. Specifically, the main learning goals include that students will be able to interpret cancer-related statistics, including incidence rate, mortality rate, and relative survival, interpret graphical representations of cancer statistics, enter data into a table and analyze data presented in a table, and discuss how social determinants of health could contribute to observed differences in cancer statistics among various populations. The module is structured with several primers that provide relevant background information on key topics, including social determinants of health, cancer, and correlation versus causation. Then the main activity of the module engages students in data analysis to uncover and understand health disparities in cancer. The primers are designed such that teachers can choose whether to include or exclude them based on their students' prior knowledge. Additionally, primer content is available in both handout and presentation formats. The images on the right show part of the primer about cancer in both handout and slide format. The first primer introduces health disparities and social determinants of health. Health disparities are preventable differences in health outcomes between two groups due to social, economic, or environmental conditions. Social determinants of health are the non-medical factors that impact health. They include the conditions in which people are born, grow, work, live, and age, as well as people's access to power, money, and resources that are influenced by social and political systems. The United States Centers for Disease Control and Prevention identifies 5 broad categories of social determinants of health: education access and quality, healthcare and quality, neighborhood and built environment, social and community context, and economic stability. The primer introduces these concepts, then has students define them in their own words and identify a social determinant of health that has impacted them or a family member. For the primer on cancer, students watch 2 short videos produced by JAX, one titled "What are DNA variants?" and another titled "What is cancer?" Then students answer questions that apply some of the terminology introduced in the videos. The second part of the primer introduces the statistics that are used in the data analysis activity. This includes incidence rate, which is a measure of how often a disease occurs in a population, mortality rate, which is a measure of the number of people in a population who die of a disease within a defined period of time, and 5-year relative survival rate, which is a measure of the percentage of people with a disease who are expected to survive for at least 5 years after their initial diagnosis. The primer on correlation and causation introduces these concepts using a brief example of growing sunflowers. For causation, students are presented with a story where they imagine they are studying the effect of nutrients on plant growth. You pot 20 sunflower seeds in identical pots with the same type of soil, put them next to each other on a windowsill, and water them at the same time each week. However, you give only half of the sunflowers a fertilizer with nutrients. You measure the growth of all of the sunflowers and find that the fertilized flowers grow faster. Since you controlled the light, water, and soil of the plants, you can be fairly certain that the fertilizer caused the difference in growth. For correlation, students are presented with a story where they are visiting a friend who has 2 different garden plots. The friend is growing sunflowers in each. You notice that one garden plot gets more sunlight than the other, and the sunflowers in that plot are taller. There is a correlation between the amount of sunlight the plants receive and their height. However, we do not know if there is a difference between the soil of the garden plots or whether the plants get the same amount of water. Since the amount of sunlight is only one possible difference between the 2 groups of sunflowers, we cannot say that the sunlight caused the height difference in the plants. After this introduction, students work through 4 short scenarios to practice applying this concept and determining if a relationship is correlative or causative. As an example, one scenario reads, you are a researcher studying the impact of water quality on life expectancy. You collect life expectancy and water quality data for towns around your state. You find that towns with higher water quality also have higher life expectancy. Students then need to determine whether this relationship is correlative or causative and explain their reasoning. Now that I've covered the primers, let's move on to the main activity. In this activity, students explore data illustrating disparities in cancer mortality rates across populations. There are two parts to this activity. In the first part, students compare overall cancer incidence and mortality rates in the United States. In the second part, students interpret graphs and tables to identify associations between cancer disparities and social determinants of health, including education, income, health insurance, and stage of diagnosis. The cancer statistics used in these activities come from SEER Explorer. The Surveillance Epidemiology and End Results Program, or SEER, is supported by the National Cancer Institute. SEER provides cancer statistics with the goal of reducing cancer in the United States. SEER Explorer is an online database that provides access to SEER cancer statistics by cancer site, diagnosis year, gender, race, age, and stage. The survey data utilized in Part 2 comes from the All of Us research program. The All of Us research program through the National Institutes of Health aims to advance biomedical and health research by collecting health data from 1 million or more people living in the United States. All of Us aims to recruit participants that reflect the diversity of the United States by keeping participation open to all and engaging participants as partners. We are very grateful to both of these programs and to the All of Us participants. Part 1 starts by having students examine incidence rate for all types of cancer for two different populations within the US. As a reminder, incidence rate represents how often a disease occurs in a population. Here, a cancer incidence rate of 500 for a specific population means that for every 100,000 people in that specific population, 500 people will develop cancer in a specific year. Questions in this part guide students in observing the data and estimating values of points on a graph to enter into a table. Next, students examine a similar graph, which looks at mortality rates for all cancer types for the same populations over the same time period. Again, students are asked to record their observations and translate some of the data points from the graph into a table. Students then use the data to identify similarities and differences between incidence and mortality rates between the two populations. Finally, students are asked to hypothesize factors that may be contributing to any correlations they observed, and to brainstorm what additional questions they have or what additional information would help them better interpret the graphs. In part 2 of the activity, students dive into data that examines correlations between social determinants of health and cancer disparities. There are 4 options for part 2, each of which focuses on a different social determinant of health and on a different cancer type or statistic. However, all 4 of the options follow the same structure, such that you could meet all of the learning goals for the activity with just one of the 4 options. This provides flexibility for different ways of implementing Part 2, including choosing an option to work through as a class, or having students work in small groups as a jigsaw activity. As an example, we will work through the data for option 4, which examines stage at diagnosis. First, students are provided a graph similar to those in Part 1, which illustrates a health disparity. In this case, there is a disparity in 5-year relative survival rates for melanoma between the Black includes Hispanic population and white includes Hispanic population in the US. As in Part 1, students are asked to record their observations and read data from the graph. Next, students examine a graph which shows the relationship between the social determinant of health and the relevant cancer statistic. For option 4, this is stage at diagnosis and 5-year relative survival. Cancer stage refers to the size of the tumor and how far the cancer has spread from its original location. Localized cancer has not spread, regional cancer has spread nearby its original location, and distant cancer has spread to further areas of the body. If the patient's cancer was not staged at the time of diagnosis, it is listed as unstaged. From this graph, students can observe that more advanced cancer stages at time of diagnosis are associated with lower 5-year relative survival rates. As in part 1, students are asked to estimate data points from the graph and enter them into a table. For the health insurance and income options for part 2, at this point in the activity, we instead provide a table and ask students to generate the graph. Otherwise, the questions remain the same for all options. This allows you to select the options that best align with either the social determinant of health that your students will be most engaged by, or by the skill you most want them to practice. Finally, students examine a graph using survey data from either SEER Explorer or the All of Us research program, showing the association between the social determinant of health and the two populations of interest. For option 4, students examine the association between stage at diagnosis and Black and white populations in the US and note any similarities or differences. To conclude, students answer a series of questions to reinforce that all of this data is correlative, not causative, and to develop hypotheses that might explain the correlations that they observed. For option 4, students may think about additional variables such as access to healthcare, socioeconomic status, access to public services, and sunscreen use, and may hypothesize that since melanoma is less common in Black individuals, doctors may not be trained to identify it as effectively, which means it may be detected later. Now that I've walked through the activities, we can see how they give students the opportunity to interpret cancer-related statistics, interpret graphs, analyze tables, and discuss how social determinants of health could contribute to health disparities in cancer. For teachers interested in implementing this lesson, we have a teacher guide full of resources. This includes teaching notes for each activity, implementation strategies, and links to supporting resources. We also have an answer key that is available by teacher request. All of the lessons and teacher resources for this module are available now on our Genetics Learning Resources page, which is at jax.org/stem-resources. The resource page contains tiles for each of our modules, and you can look for the module tile for the Health Disparities in Cancer module to access its content. The web page for the module has details about the module, including a description and learning goals, as well as tiles to download all of the module materials, including the teacher guide and student handouts. There is also a link to a feedback form that we hope you will fill out if you implement this module with your students. Now I would like to introduce the second lesson we have developed, a case study titled Precision Over Presumption. For this lesson, we hope students come away with the enduring understanding that making assumptions about an individual's racial or ethnic identities can lead to biases and potentially ineffective patient care. Specific learning goals include that students will be able to outline how a person's genetic ancestry could impact their response to medication, describe population allele frequency, analyze and interpret data for gene frequencies stratified by genetic ancestry, calculate genotype frequencies, and summarize why assumptions of racial or ethnic identity should not dictate medical treatment. In part 1 of the case study, the storyline follows a neurologist, Dr. Elena Carter, as she cares for a patient with severe migraines. Comprehension questions for this part focus on ensuring students understand the symptoms and triggers of migraine headaches. Part 2 introduces students to pharmacogenomics, the study of how an individual's genetics or genomics impacts their response to a medication. In the story, Dr. Carter wants to prescribe her patient a new migraine medication, but the dosing guide recommends different starting dosages based on the patient's reported race and ethnicity. A pharmacist informs her that the metabolism of medication is affected by variants in the gene CYP2C19. Questions for this part focus on how differences in metabolism might impact the effectiveness and dosage of a medication. Part 3 of the case study provides more details about the CYP2C19 gene, which encodes an enzyme that catalyzes many reactions required for metabolizing medications. Variants in CYP2C19 can affect how fast the enzyme works, which in turn can influence drug effectiveness or the number of side effects. Questions in this part focus on a specific variant in the CYP2C19 gene, which is a single nucleotide substitution that can be either an adenine, A, or a guanine, G. The A variant results in an enzyme that is not functional. Students hypothesize how different genotypes for this variant affect drug metabolism. In part 4, Dr. Carter decides to investigate the dosing recommendations further and finds data on the frequency of the CYP2C19 alleles from the All of Us research program. Allele frequency is a measure of the number of a specific allele or gene variant relative to the total number of alleles present in a population. Questions in this section engage students in reading data from the table. In an optional challenge question, students practice applying the Hardy-Weinberg equilibrium equation using the data. Part 5 of the case introduces data from All of Us on allele frequency for the CYP2C19 variant categorized by genetic ancestry. First, students are presented with data showing the proportion of the A allele count by genetic ancestry. Questions guide students in interpreting data from the graph. Then students are presented with a table that enables them to calculate the allele frequency of the A allele in populations with different genetic ancestries. The data and corresponding questions illustrate to students that allele frequency can vary by genetic ancestry, but that alleles are not exclusive to individuals of particular genetic ancestries. Optional challenge questions in this section again provide an opportunity for students to practice applying the Hardy-Weinberg equilibrium equation to the data. In the final part of the case, Dr. Carter's patient consents to genetic testing for the CYP2C19 variant to help Dr. Carter better determine the correct dosage. The test results indicate that the patient should receive a different dose than Dr. Carter would have prescribed if she had followed the initial dosing guidelines based on reported race and ethnicity. The case concludes by asking students to reflect on why the test result was unexpected and the dangers of assuming genotype based on reported racial and ethnic identity. This case study has been accepted for publication through the National Center for Case Study Teaching and Science Case Study Collection through the National Science Teaching Association. Once published, the case will be available through their website. There will also be an associated teacher guide and answer key, which will be available with an NSTA case study subscription. The guide contains case learning objectives, details about recommended course level, prerequisite knowledge, and curriculum integration, classroom management strategies, and blocks of analysis, which provide additional background information for teachers on the topics discussed in the case. We are particularly excited about connections between this case study and our Teaching the Genome Generation curriculum, including through other content on the CYP2C19 variant, such as our molecular lab protocols and a section of our sequence comparison module, and connections to our ancestry and variation in the genome modules because of the focus on genetic ancestry and allele frequencies. With that, I would like to thank you for your time and encourage you to reach out to the Teaching the Genome Generation team at TtGG@jax.org with any question.